Assalamualaikum warahmatullahi wabarakatuh. We're from Group 4 and we are going to present about the title, The Contribution of von Behring and Kisato in 1890. And the focus of their contribution is actually about the birth of serotherapy, and it is going to be presented by our group members, which is me, myself, Nik Asma Madiha, Nik Nur Alia, Najia, Nadia Nur Shifa, followed by Farah Nasira. Next, so this is our table of contents that we are going to present, so you can read it yourself. Next, so first we go through the introduction, which is the background of both of the scientists, which are Emil von Behring. Emil von Behring is a physiologist that is born on March 15, 1854 in Hansdorf, West Prussia. He actually started as a theology student, which is a course that are not actually related to science at that time. However, a French arranged for him to start his medical studies at the University of Berlin. In the following years, he served as a military doctor and eventually became a captain of the medical corps of a pharmacological institute. And in 1888, he was finally given a position as an assistant to Professor Robert Koch, which we know as the founder of bacteriology. So next, we have Kisato Shibasaburo, which is a Japanese physician. He is born on January 29, 1853 in Kitanosato, which is a province in Japan. He began his study of medicine at Ikagusho Hospital, and after graduation, he carried out bacteriological research at the Central Sanitary Bureau of Ministry of Home Affairs. And finally, in 1885, Kisato moved to Berlin to join the laboratory of German bacteriologist named Robert Koch. So, long story shortened, there he finally met with his fellow friend Emil von Behring and managed to develop the first effective therapeutic serum against diphtheria and tetanus. And their discovery gave rise to the newer vaccination program that we currently have now. So the journey of them developing the therapeutic serum will actually be presented in detail by the next presenter.
My name is Najia Binti Kamaluddin, and I will proceed with the experiment. Long before the 20th century, scientists have suspected that blood was involved in immunity against pathogen, but there is no experimental evidence for this. So in 1890, Kisato and von Behring performed an experiment to prove this statement by immunizing guinea pigs against diphtheria with blood products from animals that had recovered from the disease. There were many diphtheria and tetanus cases at that time. The picture shows their experiment, which consists of experiment A, B, and C. In experiment A, guinea pig 2 was injected with serum from unexposed guinea pig 1, but it resulted in diphtheria. In experiment B, guinea pig 1 had been injected with toxin from bacteria strain A, which then developed in its blood serum. Then guinea pig 2 was injected with the serum from guinea pig 1, but it was not suffered diphtheria. Experiment C also worked in the same way like experiment B, but it used different toxin B. Later, guinea pig 2 also had no diphtheria. In conclusion, the response of the guinea pig 2 in experiment B and C to the diphtheria toxin is an example of adaptive immunity. After exposure to the toxin, they made a protective factor. Moreover, the immunity was specific. Serum of toxin exposed guinea pigs is protective against later exposure to toxin from the same genetic strain of bacteria. Based on the animal model, Behring realized that serum protection might work for human diseases as well. It did. Next slide. Later, the agent of this immunity was identified as an antibody protein. And the process of acquiring immunity from antibodies received from another individual was called passive immunity. Pass to the next presenter.
I will present about the passive immunity, which is the concept that has been used by Kisato and von Behring. So, passive immunity is the process when a person receives antibody for specific disease that are not from their own immune system. So basically, there are two types of passive immunity, which are natural and artificial. Natural immunity is, for example, that an infant receive antibody from their mother through breastfeeding. But particularly in this topic, we are talking about artificial passive immunity, which are antiserum. Clostridium tetani and Corynebacterium diphtheria are bacteria that are responsible for causing tetanus and diphtheria disease. Both bacteria produce toxin that can give harm to our body. So, to treat these diseases, the scientists produce antitoxin, which can neutralize the toxin. So, how the antitoxin were made? So first, the scientists will grow, for example, diphtheria-causing bacteria in the laboratory and harvest its toxins. Next, researchers or scientists will inject the horses with the diphtheria toxin. As for your information, horse is immunized towards diphtheria. So, the horse are able to produce diphtheria antitoxin. So, scientists will collect the blood from the horse and spray out the antitoxin rich serum. Then, it will apply it to treat people. Go to the differences between active immunity and passive immunity. Passive immunity are actually antibody from other organism, different with active immunity, which are produced by our own body. Active immunity long lasts, but passive immunity is short lasting. It is because passive immunity does not have immunological memory like active immunity. So that's all from me. Thank you.
Alright, now let's move on to the development of diphtheria antitoxins for humans. So diphtheria is a very serious infection caused by strains of bacteria called Corynebacterium diphtheriae that make poison or toxin mainly in the upper respiratory tract. It can lead to difficulty of breathing, heart failure, paralysis, and even to the extent of death. So we can see here in the picture below, there are thick grayish fibrin membrane that forms over the site of the infection as a result of the bacterial growth. Therefore, to overcome this problem of diphtheria, antitoxin was introduced and developed in the late 1800s. Diphtheria antitoxin is a medication made up of antibodies used in the treatment of diphtheria, and it is given by injection into a vein or muscle. This antitoxin was made from blood plasma of horses that have been immunized against the diphtheria toxin. Now, based on the data here, we can see a very serious number of cases of this disease being eradicated ever since the vaccination was introduced, which was known as DTP3 vaccine.
Okay, so for the conclusion, next slide. I will be presenting about the von Behring and Kisato contribution to the modern world. So the first antitoxin to diphtheria was discovered by Emil von Behring and Shibasaburo Kisato in 1890. And Emil von Behring received the Nobel Prize in Physiology or Medicine 1901. And today we can see that antitoxin is widely used to treat botulism, diphtheria, dysentery, gas gangrene, and also tetanus. Next slide. So Emil von Behring also and other researchers showed that by means of blood plasma or serum, antibodies could be transferred from one person or animal to another person, who also then become immune. And in 1900, Emil von Behring introduced serum from immune horses as a method to cure and prevent diphtheria. Next slide. As for the Islamization, there is a hadith narrated by Abu Hurairah radhiyallahu anhu. Prophet Muhammad sallallahu alaihi wasallam stated that whoever relieves a believer of some of the distress of this world, Allah will relieve him of some of the distress of the day of resurrection. And whoever comes to the aid of one who is going through hardship, Allah will come to his aid in this world and the next. Sahih Muslim. And there's also a verse from Surah Fatir, verse 28. It is only those who have knowledge among his slaves that fear Allah, which we can apply in our daily life, that through seeking knowledge and using them to help others, we can deepen our faith towards Allah Subhanahu wa Ta'ala.